Yeast metabolism is central in beer making and wine making. By the way, have you ever thought of this question? Who discovered wine? Or who discovered beer? Story has it that there was a man who found a honeybee in a closed jar. He drank the liquid, found it to taste funny, and his head started spinning. He laughed for no reason and he felt powerful. So we know that he is starting to get drunk. He drank all of the liquid, and the following morning, he was having headaches, coupled with dizziness and an unpleasant taste in his mouth. You know it. He experienced a hangover. Archaeology has discovered jars containing remains of wine that are 7,000 years old and could be very likely that humankind's first encounter with alcoholic beverages was by accident. So, in the course of human history, several civilizations began producing fermented beverages. You have the Greeks, the Chinese, the Egyptians, Vikings, and they used different sources such as grapes and grains or even fruits, other fruits. And at that time, what they only knew is that living fruits and grains in covered containers for a long time produced the fermented beverages, the wine and beer that we know now. They named the process fermentation from the Latin word fervere, which means to boil. That is because they observed that these mixtures of crushed grapes kept in large vessels produced bubbles as if it were boiling. And through a system of trial and error, they were able to improve the process. They knew that if they didn't keep the mixture for too long, there will be no alcohol produced. But if they kept the mixture incubated for a long period of time, they would find a rotten mixture and an undrinkable liquid. They also learned that temperature and air are important factors to fermentation. And probably you've also seen videos of people who use their feet to crush the grapes that they use to create wine, probably in some movies that you have seen. They indeed perform this, and in so doing, they transferred microorganisms from their feet to the mixture. So, all of these knowledge, old knowledge about fermentation. But it's your chemists who discovered the principle, combined with the development of powerful lenses used in microscopes. You all know who invented the microscope: Anton van Leeuwenhoek. Not exactly the microscope, but he was able to create powerful lenses, so magnifying lenses. And this transported him into a whole new different world, the world of your microorganisms. And he used his lenses to look at literally everything. And thus, he was the first person to view your yeast through his lenses. So he saw them as globules floating in the fluid, but he thought that they were merely the starchy particles of the grain used for fermentation. In the 18th and 19th centuries, we have your chemists working to understand the process of fermentation. So Antoine Lavoisier, he's famous for his law of conservation of mass during chemical reactions. He also actually studied fermentation. And his studies on fermentation also added proof to his law of conservation of mass. Through careful and accurate measurements possible during his time, he was able to conclude that a starting batch of sugar, after undergoing fermentation, would be converted into two sets of products. That means there were two pathways, or that there were two processes taking place. So, from the original batch of sugar, two-thirds of that would be converted to alcohol. So, it would be reduced to alcohol. and the remaining one-third would be oxidized to carbon dioxide. He even went on to say that it was possible to combine alcohol and carbon dioxide in the right proportions to produce sugar. And through what we have learned, that is impossible because you would need enzymes or some other catalyst. However, he still was not able to make the connection between the yeast and the fermentation process. So enter a French inventor who, using a more powerful microscope, more powerful than the lenses of Leeuwenhoek, he was able to observe that during alcoholic fermentation, yeasts undergo budding. And they already know that budding is a mode of reproduction in organisms. So with this observation, they were able to establish that yeasts are living organisms and that they are one-celled organisms. So now that yeasts have been established to be living organisms, enter Pasteur. So we all know the works of Pasteur. When we discussed the history of enzymes in connection to the vitalism theory, Pasteur was mentioned. So his works on yeasts led him to conclude that only living yeast could transform sugars into ethanol. And we know that that is not true, right? So there was another guy after him, Edward Buchner, and he was the one who was able to show that even without the living yeast, as long as you have the yeast extract or some components extracted from yeast, you can still ferment sugars to alcohol. So going back to Pasteur, his careful experiments led him to discover that there are other products of fermentation. So it's not just ethanol that is produced in fermentation. So there are other compounds such as glycerin, succinic acid, among others. Pasteur also provided proof that fermentation is a consequence of yeast multiplication. And of course, his very important observation is that this process of fermentation happens in the absence of oxygen. So it's an anaerobic process. And then finally, after performing a consultation work for a guy who owns a distillery, Pasteur discovered that yeasts are not the only microorganisms that can perform fermentation. Thus, they were able to say or conclude that there are two types of fermentation. One is alcohol fermentation, and the other is lactic acid fermentation. And his observations um, told them that this lactic acid fermentation is caused by organisms that are smaller than yeasts. And eventually, they established that this lactic acid fermentation is performed by bacteria. So in the context of winemaking, alcoholic fermentation can be broadly classified into the metabolism of carbon, nitrogen, and sulfur compounds. Of course, carbon metabolism is the prime pathway, and we will go to this in a while. Ultimately, the amount of alcohol produced depends on the starting amount of your sugars in the source, such as your fruit, for example. So during this process, a lot of organic compounds, particularly acids, are synthesized or degraded by the yeast. Understanding the various pathways that yeast can undergo in fermentation and the products produced in each pathway is vital for beer production and winemaking, because there could be some products that could affect the quality of the taste of your wine, or if there are products that could improve the taste or the scent of your wine. That is why understanding yeast metabolism, the pathways, and the products is very important in these industries. So I mentioned that the primary metabolism pathway that will be mentioned in this lecture video is carbon metabolism. So a limited number of carbon compounds can be used by your Saccharomyces cerevisiae the most common species of yeast used in fermentation. So they can use monosaccharides such as your glucose and fructose. They can also use disaccharides such as sucrose and maltose, and some trisaccharides such as raffinose. Pentoses, like your ribose, are not utilized by your S. cerevisiae. I mentioned before that in carbon metabolism, the yeast needs pyruvate, or I think this is the first time I'm mentioning this. The yeast needs pyruvate in fermentation. So, of course, this will be supplied by your ubiquitous glycolysis or your glycolytic pathway, or the other less common term for glycolysis is the Embden-Meyerhof-Parnas pathway. So the pathway is attributed to three guys who was working in some institute in Germany. They were the ones who were able to elucidate a lot of mechanisms in the glycolytic pathway. So you have Embden, Meyerhof, and Parnas. So that's the Embden, Meyerhof, Parnas pathway, or the EMP pathway. So that's the less common name for your glycolysis. So glycolysis takes place both in the fermentative and respiratory modes of metabolism in all organisms. And you know that glycolysis involves 10 steps, and it consumes a net amount, or it produces a net amount of 2 ATP molecules, 2 NADH molecules, and it consumes 1 molecule of glucose. So what I want to say is that from 1 molecule of glucose, after it goes through the glycolytic pathway, you get 2 molecules of ATP, 2 molecules of NADH. In yeast, under favorable conditions, and of course in the presence of oxygen, pyruvate can actually enter the citric acid cycle in the mitochondria. So this allows for the production of 36 ATPs. However, in the absence of oxygen, the electron transport chain cannot operate. So this will leave the yeast cell with an overload or oversupply of pyruvate molecules and NADH. So the cell needs to oxidize the NADH molecule so that it will have this steady supply of NAD. So that even if the pyruvate is just there, it will continue to harness sugars from its environment. And as long as it has NAD, then it can at least get 2 molecules of ATP from each molecule of glucose. So this is possible through fermentation. So fermentation is defined as an anaerobic metabolic process. So anaerobic meaning it occurs in the absence of oxygen. This does not only take place in single-celled organisms, take note. So we know, or I think you are familiar, that fermentation also takes place in our muscles, for example, as we shall see in a few slides. But wait, it is important to note that it is possible for other microorganisms to undergo respiration in the absence of oxygen. So that's what we call anaerobic respiration. So they can still use the electron transport chain even without oxygen. How is that possible? They use a different electron acceptor. So recall that the role of oxygen in the electron transport chain is the final electron acceptor. So it's converted to water. So there are microorganisms that instead of using oxygen, they evolved to use, for example, sulfate ion as their final electron acceptor, nitrate ion, or simply the sulfur compound, the molecular sulfur, and so on. So Most of these microorganisms are prokaryotes. So they are bacteria and your archaebacteria living in oxygen-deprived environments. So here you see an aerial photograph of a coastal water, and you see the green region here in the sea. This one is an indication of an overgrowth of sulfate-reducing bacteria. So going back to fermentation, if pyruvate cannot enter the citric acid cycle due to the lack of oxygen, what will happen? There will be a buildup of your NADH. So all of your NAD will be consumed and converted to NADH. The yeast, when it lacks NAD, it can no longer metabolize the glucose molecules from the environment. So it doesn't have any supply of energy now and it dies. That is why it switches to fermentation, because fermentation, in fermentation, after glycolysis, you have additional reactions, and their purpose is to regenerate the NAD. So this is made possible by your NAD dropping its electrons to another molecule, such as an organic molecule. So we have two most common and well-known types of fermentation. We already know them through Pasteur. So one is the lactic acid fermentation, which again is primarily performed by bacteria. So, not only by bacteria, but also our muscle cells. So this happens in our muscle cells when we are performing strenuous physical activities. In this type of fermentation, your NADH drops its electrons directly to pyruvate. So NADH floating around inside the cell sees an abundant supply of pyruvate and thinks, hey, maybe I should drop my electrons, my baggage of electrons to those pyruvate molecules. And that's what happened. So the pyruvate molecules, after accepting these electrons from the NADH molecules, they are converted to lactate. So your NADH, after dropping their electrons, they are converted back to NAD. And now the cell, again, can use the regenerated NAD to harness energy from the glucose molecules through the glycolytic pathway. So when you exercise hard, for example, the supply of oxygen to the muscles cannot keep up with the demand. So they undergo lactic acid fermentation. So before, a lot of people thought that the lactic acid produced causes muscle soreness. But they are thinking otherwise now, because what happens to the lactate produced in your muscle cells, they get transported to the liver and they are converted back to pyruvate molecules. And these pyruvate molecules can then um, be metabolized in the most common way possible, which is the citric acid cycle. Your red blood cells also undergo this type of fermentation because they don't have mitochondria. And the second type is alcohol fermentation or alcoholic fermentation. This is also known as Neuberg's first form of fermentation. Carl Neuberg is not actually popular for some people, but he is considered as the father of modern biochemistry. So he was also part of the group of Emden, Meyerhoff, and Parnas. So he was also exploring these metabolic pathways. So we have that another classification of fermentation. So alcohol fermentation is Neuberg's first form of fermentation. Unlike lactic acid formation, wherein you have only one additional step to glycolysis, in alcohol fermentation, you have additional two steps. So in the first step, you have pyruvate converted to acetaldehyde. In the process, you release a carbon dioxide molecule. So pyruvate is essentially decarboxylated. So what do you think is the name of the enzyme for this process? If you guessed pyruvate decarboxylase, then you are correct. So this is the enzyme involved in the decarboxylation of pyruvate to acetaldehyde. And then your NADH molecules drop their electrons to the acetaldehyde molecules. So the product is your ethanol. And then the cell now has regenerated NAD molecules and again can continuously harness energy from the sugars. So for alcohol fermentation, what you get are two molecules of ATP, two molecules of ethanol, and two molecules of carbon dioxide. And your cell gets its constant supply of NAD molecules. By the way, for the second step, there is also an enzyme that catalyzes this second step. I mean, the second additional step. It's alcohol dehydrogenase. So you have pyruvate decarboxylase and alcohol dehydrogenase in alcohol fermentation. So this type of fermentation, and I'm referring to Neuberg's first form of fermentation, this is favorable at pH 7. Surprisingly, they observed that at higher pH values and in the presence of excess NAD molecules, the reaction tends to proceed backward. Meaning, so for example, you have a batch of yeast molecules that already produced ethanol, and then you suddenly switch the pH of the system to 7 and introduce an excess of NAD molecules. What the yeast cells will do is to convert ethanol back to pyruvate. And from my reference, they didn't mention the mechanism why the yeast cells would do that. Since we have Neuberg's first form of fermentation, we also have the second and the third forms. So the second form of fermentation, which would be called the glyceropyruvic fermentation, is also your alcohol fermentation, but this one takes place at pH 6 or below. So it's in the acidic pH range. And what happens here is that they observe that they only get small amounts of glycerol in this second form of fermentation and other byproducts. Now, the second form of fermentation was of interest to scientists during World War I and World War II, and also during the interval of those two wars, because this type of fermentation produced glycerol, and they wanted lots of glycerol. So aside from using glycerol as the coolants in the vehicles, they also used glycerol as components of explosives during those wars. That is why they studied this second form of fermentation a lot. So they noticed that they can get glycerol from this second form, but only in limited amounts. So what they did is they explored how to maximize the amount of glycerol that they can get from the second form of fermentation. And what they observed is that they can enhance this by adding sulfite. So what happens is that the sulfite depletes the acetaldehyde. So it becomes unavailable for conversion to ethanol, and NADH accumulates. So unless other hydrogen acceptors are provided, the cells will die because the NADH molecules do not get converted back to NAD molecules. Now, this actually can also happen if there is an absence or low activity of pyruvate decarboxylase, or if there is a high activity of aldehyde dehydrogenase. This one is under alkaline pH. So this one, they observed that if they added sulfite, they can actually increase the amount of glycerol that can be obtained from the second form of fermentation. So we will not go into the detail, into the pathway, the specific pathway that the yeast takes when it switches to the second form of fermentation and you add sulfite. Okay, so there's probably some enzyme that maybe uses sulfite as a cofactor, and then enhances the conversion of your organic molecules there, maybe the acetaldehyde enhances the conversion to glycerol. So we will not go into the details. And then we have Neuberg's third form of fermentation. This one takes place in alkaline conditions. So what happens here is that you have the dihydroxyacetone phosphate, which is one of the intermediates in glycolysis. In this form of fermentation, NADH drops its electrons to this molecule, your dihydroxyacetone phosphate. So you have your NAD regenerated, and you produce glycerol 3 phosphate. This glycerol 3 phosphate gets dephosphorylated, so you remove the phosphate group and you get glycerol. So again, the third form of fermentation is another thing that they studied extensively because it also allowed them to produce glycerol. So for the first form of fermentation, which produces ethanol, okay, uh, this yield, ethanol yield in alcoholic fermentation depends on the ethanol tolerance of your yeast. So the ethanol produced in alcohol fermentation is actually toxic to your yeast cells. Ethanol can inhibit the enzymes involved in the catabolism, and it can also interact with the cell membrane of your yeast cells and eventually destroy the cell membrane. So this leads to leakage of your cell contents and of course, death. And they also found out that the tolerance of your yeast species depends on their membrane fluidity. So we just talked about membrane and its properties in the previous module. So probably if your membrane is less fluid, then it can be more tolerant to ethanol. But if it's more fluid, then it's less tolerant to ethanol. So a wide range of yeast species can tolerate 5% by volume to 21% by volume ethanol. And this is actually the main reason why beer and wine, which are produced through alcoholic fermentation or the Neuberg first form, have a percent volume of alcohol within this range because you cannot increase the percentage of your ethanol, otherwise the yeast cells would die. In other cases, if you want to produce liquors, so those beverages with higher concentration of alcohol, what they do is they distill the alcohol so as to maintain a minimum concentration of ethanol in the mixture, and then maybe they just add the distilled ethanol to the final liquid afterwards. Regulation. So I will mention a few things or a few information on how fermentation in yeast is regulated. So there are two major points of control in fermentation. So the first one, and this one is the one that they really observe a lot, is the transport of sugar across the cell membrane. So this one, if the transport is fast, then the rate of fermentation is also fast. If the transport is low, then the rate of fermentation will also be slow. And then the second point is phosphorylation of the sugar. So once you have your sugar molecules inside in the cytosol, then the rate of its phosphorylation also determines the rate of fermentation. Furthermore, loss of enzyme activities in steps 1, 3, and 10 of glycolysis had an immediate impact on glucose transporters, hence affecting the rate of fermentation. So do you remember the first, third, and last step of glycolysis? First step is the phosphorylation of the sugar, and I mentioned that it's one of the major points of control. The enzyme involved there is hexokinase. The third step, the enzyme for the third step is phosphofructokinase. And the last step pyruvate kinase, right? And another important observation that they made is that when nitrogen is limited, for example, the ammonia present in your reaction mixture, transporter activity also slows down, or sometimes it's lost completely. Hence, fermentation will also become slower, or fermentation will completely stop. Aside from these, there are also some effects that are common in fermentation. So I think you already know or you are familiar with some of this. So the first one, which is the most common, is the Pasteur effect. So the Pasteur effect, from the name itself, was discovered by Pasteur. So what he observed is that once you introduce oxygen to a batch of yeast cells that are undergoing fermentation, you will observe a decrease in glucose consumption. So his conclusion is that oxygen, oxygen suppresses glycolysis. So the presence of oxygen there will lead you to think that the cells are under less cells are undergoing glycolysis, or the rate of glycolysis became slower because the rate of glucose consumption decreased. Now that might be the case during Pasteur's time because that was his observations. However, more scientists studied this Pasteur effect and they realized that the Pasteur effect can only be observed under special conditions. So it cannot be observed all the time. So unless you are under those special conditions, that's when you will observe the Pasteur effect. So the specific conditions that they established to be important for you to observe the Pasteur effect is number one, steady growth rates, and number two, resting cell states. There. Now, the second one is the Crabtree effect. So, the Crabtree effect can be classified as short term or long term. In the short term Crabtree effect, you have a steadily cultured system. And what you do is you suddenly increase the concentration of sugar in the cultures in your culture. And what you will observe is that even in the presence of oxygen, the yeast cells will still undergo alcoholic fermentation. So it's like the high concentration of sugar this time suppresses uh what do you call this? Glycolysis. Okay? Now, in the long-term Crabtree effect, you have a steadily cultured system at high growth rates. So they will also display aerobic alcohol fermentation once you increase the concentration of the sugar in the system. So you see the difference. In short term, you suddenly increase the concentration of sugar in your culture, but in the long term, you have a steadily cultured system with a very high growth rate, and then you try to increase the sugar concentration and observe it for a long period of time, and you will see that even in the presence of oxygen, it will still undergo alcoholic fermentation. So it's called aerobic alcohol fermentation because oxygen is still present, but the cell chooses to undergo uh, alcohol fermentation. Now, there is supposed to be another slide here, and it's about the Custer effect. I guess I was not able to save that version of the PowerPoint file. But anyway, the Custer effect is the reverse of your Pasteur effect. So in the Pasteur effect, if you increase your oxygen, the yeast cells slows down their glucose consumption. Now, in the Custer effect, if you increase your oxygen, the concentration of oxygen in the culture, what you will observe is that there is or there will be an increase in the rate of fermentation. And this means that there is also an increase in glycolysis. So what happens here, this is not displayed by your Saccharomyces cerevisiae or other species of yeasts. So this Custer effect is displayed by a different genus of yeast. So this is all about the redox balance. So the redox balance being talked about here is the ability of the yeast to immediately regenerate the NAD molecules. Okay, so sometimes, so in the Custer effect, sometimes the rate of the regeneration of NAD molecules cannot keep up with the demand for the energy. Hence, uh, what the what the cells do is to compensate for that by continuously harnessing the energy from the glucose molecules, and hence even if there is an increase in the oxygen, in the amount of oxygen, which could have been a signal to revert to the citric acid cycle, what the cell does is that it just continues to get the glucose molecules to undergo the glycolytic pathway. So that's your Custer effect. And so the discovery, this is the end. The discovery of yeast metabolism is just one of the milestones in human history that proved a lot of things. Number one, science can answer the most curious questions. And number two, collaborations can reach even greater heights. So you've seen how chemists, biologists, those who invented lenses, so physicists, they all combined their discoveries, their knowledge, their observations, and it led to this extensive body of knowledge about yeast metabolism that is used in several industries. And not just in beer making and wine making, but also in um, organic synthesis, for example. So some of them get their starting molecules from the byproducts of fermentation. So I hope that this inspires you to be more curious and to be more driven to find answers to your questions. And you have your DIY activity on yeast metabolism. Some of you already submitted their selfies with their setups, and I found their setups to be quite revealing about yeast metabolism. So, what did you find out? If you have more sugar, will there be more fermentation? If you don't have oxygen, will there be more fermentation? So I'm excited to read your outputs for this module.